cell culture system. So where you see in Vero cells, in panel A, uninfected cells and the infected cells. The cells show a lot of cytopathic effect. And also we could actually visualize these infected cells in the panel B, see using SARS-CoV-2 specific antibodies. The red labeled cells are actually infected. We use 2 different antibodies just in case we want to make sure the infection is real. Both antibodies are specifically picking up the viral replication. Then this is where our collaboration with Robert in the shared resources facility, where we started setting up a medium throughput drug screen. So for that, we want to have established some of the controls needed for this study. And panel C, you could see Damoiseaux, that's a vehicle alone. There are a lot of virus replication happening.
Excuse me, Vaithilingaraja. Your slides aren't being shared. Your slides
Are being shared?
not being shared.
Oh, not being shared. Oh, okay, okay. Let me get that.
Yes.
Okay, got it. So just started. So
Thank you.
okay, so let me recap quickly. So we got the viral strain from CDC in mid-March, and we established the system. The panel A, using Vero E6 cells, we could actually expand the virus. Where here you see the uninfected culture. With the infection, the cells are actually showing a lot of cytopathy, those positive cells. And also we verified with SARS-CoV specific antibodies. One is detecting spike, the other one is all the structural proteins. Both are specifically picking up the viral replication. So here, this is where we established collaboration with Robert, and for setting up a medium throughput drug screen, where we established various controls, where we included hydroxychloroquine. This drug does work, at least in the cell culture condition, where it blocks the virus entry, so the virus cannot replicate it. The other drug is already in the clinical trial, that's the interferon beta, that also efficiently blocks the replication. So this study requires biosafety level 3 containment. So UCLA has uniquely positioned to have this running facility. So as soon as this outbreak came, we were all prepared to handle this threat. So very quickly, we established various assay systems. And I just want to share a few of the slides from our recent study using cardiomyocyte infection. So you all know this COVID-19, the SARS-CoV-2 has people with underlying cardiovascular disease to have higher risk of morbidity and mortality. This could be due to the virus replicating directly into the cardiac cells, or due to the low oxygen level they are getting because of the lung system is affected, or could be inflammation. So we want to see whether the virus can actually replicate in heart cells. So we used this human system. It's an induced pluripotent stem cell derived cardiomyocytes model system. This work actually with a close collaboration with our colleagues in UCLA Medical Sciences, and also his research fellow, Dr. Sharma. So they had this beautiful cardiac cardiomyocyte cell system, and we use this to infect. So essentially, here, this is kind of a summary of a few weeks of work, where we, in uninfected cells, this is a cardiotroponin marker, and without infection, you don't see that infection marker. However, with the infection, you have a lot of virus replication happening. And also, when there is that much replication happening, the cells are undergoing apoptotic death. So we use the cleaved caspase 3 to stain that dying cells. So this tells us that virus can directly replicate in this cell system, but still, there's a long way to go before to make a clear conclusion. But at least it is giving us a way entry point to study the effect of this virus on cardiovascular system. Here, essentially to show, we looked at different replication compartments inside the cell using the spike antibody staining and also viral genome staining. So we see a beautiful replication in this cell type. And we quantified various infection markers, apoptosis, and also viral markers. It's highly enriched during the infection. So so I was not surprised to see this result. It is essentially like any virus infection you could see. But the striking feature is here in the cardiac cells, this uninfected cell, what you see is the pulsating heart. It is pulsating in a kind of 12 to 15 beats per minute. However, upon infection, it just there's no pulse. The heart stopped beating. So that is a kind of a very striking feature, which we got very excited about. And also, we could actually use the system kind of drug testing or more understanding the molecular cellular basis of this COVID-19. So with this model, we also, in parallelly working with my other colleague, Dr. Brigitte Gomperts, on lung model system, which kind of nicely, connects all the dots. And I invite my colleague to, continue. Thank you.
Thanks, Vaiti. Thank you. Great. Well, my name is Brigitte Gomperts, and I'm a professor of pediatrics and pulmonary medicine here at UCLA. And I'm a physician scientist, and we've been working on lung repair and regeneration for many years. And over the last decade or so, we've started to build primary human lung models, particularly to study a number of different lung diseases, because there's been just a paucity of lung models in the field. And it's these lung models that we're now repurposing to study COVID-19, along with Vaiti and Robert. And so that's what I wanted to share with you a little bit today, is about some of the models and how they're being repurposed. So next slide. So the lungs are a very complex structure, both anatomically and functionally, and we have to think about them as starting really in the upper airway, in the upper respiratory tract, with sinuses and throat, and all the way down to the trachea. And it's really this upper part of the airways that functions for host defense. And this is through mucociliary clearance. And so the mucus cells produce mucus, which traps viruses, bacteria, fungi, and all the pollution that we're breathing in. And then the ciliated cells act with these hair-like projections to beat unilaterally to move the mucus up and out of the body, and in that way protect the lungs. There are then the bronchioles, which are primarily there to conduct oxygen and carbon dioxide. And that then extends down to the little alveolar structures, which form this tremendous, very large surface area. There are billions of these alveolar sacs, and they are surrounded by type 1 and type 2 alveolar cells, which together with the endothelial cells create the respiratory membrane across which the critical gas exchange occurs. And so, as we all know, SARS-CoV-2 virus is an acute respiratory virus, and it can really infect anywhere along this tract. But when we think about disease modeling, we have to think about all the different cell types and about the different regions of the lung in terms of where the infection occurs. Next slide. And so this slide just highlights the different histologies of the regions of the lung, and also highlights how if we want to model the disease, it's going to look very different depending on the regions. So in the trachea and bronchi, there's a pseudostratified columnar epithelium and the very important submucosal gland structures. And moving down, we have the bronchiolar epithelium, which is columnar to cuboidal and leads to the bronchoalveolar duct junction. And from there, we have the alveolar structures where the type 1 and type 2 alveolar cells are. And it's the alveolar type 2 cells that have classically been shown to have a lot of ACE2 receptor expression and have been, of a lot of interest, because obviously with COVID-19, we're seeing a lot of very severe lower respiratory tract infections. And the disease has been, we've already seen a lot of diffuse alveolar damage and acute respiratory distress syndrome, where there's been a lot of inflammation, edema, and destruction of these alveolar units, which has caused a lot of problems with hypoxia, gas exchange, and really has resulted in these patients becoming so sick. Next slide. And so I just wanted to highlight two different regions that we're modeling right now. One is with the air-liquid interface culture system. This is a very well-known system. It's been around for more than a decade, and it involves a transwell membrane system where the basal stem cells are plated, and once they form a monolayer, they then differentiate into the mucus and ciliated cells of the airway epithelium. Typically in our cultures, we can push the differentiation either in a mucus towards a mucus phenotype or a ciliated cell phenotype. We can get up to 80% of the cells being ciliated, or we can shift the balance towards more mucus production. We use primary human cells that we have from normal donors as well as from patients with a number of different illnesses. And we see differences, of course, across different patients, which is very interesting to us as well. We've also developed with some bioengineering strategies, what we're calling an ALI 2.0 system, where it's not just about the epithelial cells, but we're able to introduce other cell types that are found within the proximal airway. We're able to recapitulate some of the mechanotransduction forces that are seen in the upper airway, as well as introduce some inflammatory cells. And so this is a novel culture system that is yet we haven't published on, but I think is very interesting and could be also very useful with this setting. Obviously, we're infecting these cultures and seeing very interesting phenotypes and studying. Obviously, it's a wonderful tool to study biology, as well as very good for drug screening too. And all of this, of course, is in collaboration with Vaiti, who's infecting our cultures. The other model that we're doing is the distal airway alveolar sac modeling. It turns out that type 1 and type 2 alveolar cells are actually very difficult to culture. They really require additional cell types and really recapitulation of their microenvironment to be able to grow. And so we've created a scaffolding. Essentially, it's a bead-based inverse opal scaffold, which has created a co-culture system. And in this way, we recreate that very large surface area. We have type 1 and type 2 cells. We're also able to culture with endothelial cells, inflammatory cells, mesenchymal cells, and the two images, I don't know if I can point, I probably can't, but the image on the left next to the micrograph, which shows the interconnecting alveolar spaces of the lung, is showing you what our model looks like, and the immunofluorescent image on the right is showing you a cross-section through actual human lungs. So we really very nicely recapitulate those alveolar spaces and the different cell types that we find within the alveolar structures. So we can just maybe move on to the last slide. So this is just some examples of some of the screening that we've been doing and some of the directions we're going in. In conjunction with Steve Jonas, a bioengineer on campus, we've developed a system to do aerosol delivery to the air-liquid interface cultures, and we can do multiple different aerosols to different ALI cultures. And so this is something that we're very excited about, because, you know, obviously the idea of aerosolizing some drugs to the lungs is attractive for some scenarios. And then we're also doing high throughput imaging of the lung organoids. We can actually get them into a 384 well plate system, and we can use things like high throughput confocal microscopy, which is shown with the images on the right, the little red organoids shown in the top panel, and then on the bottom panel is just an example of the 96 well plate with organoids under bright field microscopy. And so all of the screening is done in conjunction with Robert Damoiseaux. So I'm going to hand over to Robert so he can talk more about his screening core.
Awesome. So let me share my screen here. Let's see. Sharing just the wrong screen here. Okay. So is that visible? Okay.
Yes, we can see it. Thank you.
Great. So what I do at the University of California, Los Angeles is I run, I would say, something that's a little bit like a small biotech within a university. So we've built up capabilities to perform drug discovery, as it were, as you would find it at Novartis. And scientifically speaking, that's precisely where I grew up. I grew up in industry, and my focus has been really towards bringing the capabilities of what's called high throughput screening to the university, and also making this available for the people around Los Angeles, for the biotech, for the even big pharmas that we have around here. So that's what I've been up to in the last 15 years. So why do I put so much emphasis on that? Because high throughput screening is basically the way to make your research much more efficient. And if you, for example, compare here, I have a little image here on the screen. You have a laboratory from 1900 and you have a laboratory from 2000. You see that they're almost identical in terms of layout, number of people. Arguably, the people around 1900 are a little bit better dressed, but that's where it stops. The productivity is almost the same. So when you now go in and you compare that to my laboratory, as it were, you'll see that there's almost no people, but there's a lot of equipment. So high throughput screening, by and large, is defined as screening the same assay or performing the same assay more than 10,000 times in a single day. And what this means for you is that you are able to execute what you normally do one at a time, tens of thousands of times here at UCLA in a single day. So if we are looking for drugs, that's really the way to go. And that's what we're also bringing to bear on, for example, COVID-19. There's many different modalities here, and as you can see, HTS has made quite an evolution. Started out with really simple target-based screening in the 80s and 90s, moved into cell-based screening with the completion of the Human Genome Project, where we now had all targets accessible. Then later on moved into functional genomics, where we're studying now the interactions of proteins within a cell, or, for example, cell-virus protein interactions. And then that's precisely what you've seen here, what Brigitte has been showing you, complex physiologically relevant systems that really mimic very well how different cell types interact. And that's where you need really advanced capabilities like high content screening, high content screening paired with confocality and enhanced image processing and so on. So that's where these things are going. So tiny bit about our resources here. We have about 3,600 square feet of BSL2 plus approved space. We have about 200,000 small molecules. They are arranged into different libraries, starting with chemical genomics probes that you use instead of traditional mutagenesis, where you are able to knock out key enzymes or pathways within the cell to probe, for example, whether a certain pathway is necessary for viral replication, in our case here. FDA-approved drugs, of course, you want to check if any of the approved drugs is already working against the virus, and natural products, targeted libraries against high-value targets like proteases, kinases, GPCRs, ion channels, and so on. And then you're going into lead-like diverse and smart libraries. That's augmented in our case here with about 300,000 functional genomics probes. We have genome-wide CRISPR libraries, arrayed, pooled, lentiviral cDNA libraries. So for example, if you have a cell line, if you're wondering, how does this cell line do with viral infection, but it doesn't make ACE2, you could, for example, reintroduce that using our cDNA library, very, very simply. So that's something that we do quite a bit these days. And I would encourage you to check out the website, mssr.ucla.edu, to see what resources we have. And we also have capability of actually providing some of these particular resources to you as researchers. So a little bit about the equipment that we have here. A lot of it, well, maybe I should just start out with my favorite toy. That's the automated confocal spinning disk microscope. And that's also what Brigitte has shown you just a little bit earlier. That takes confocal images of 3D4 well played in about 20 minutes. And the data are getting screened real time into a database, evaluated, and so on and so forth. Of course, if you're doing high throughput screening, you don't want to stand there and do this by hand. So there's robotic arms that you see here in the background that loads, unloads this, and also, for example, loads a plate reader, but it's loaded from here, from the automated incubator that has over 200 plates in it. And then on the left-hand side is our plate production system, automated liquid handler, flex station for assay development, another incubator, and so on. So on top of that, we also have large particle sorters for everything from zebrafish to solid support chemistry beads. It's a very, very complete set. We work with a lot of different entities here on campus. That's just what we have locally here. We work a lot with the CTSI. We work a lot with CIN. It's the Center for Environmental Implications of Nanotechnologies. We are a facility for the Johnson Comprehensive Cancer Center. So we have also huge interest in cancer in addition to infectious diseases. And we are housed on the lower right-hand side. You see it in the CNSI. We also have a huge interest in nanotechnology. So we've been leveraged so far for a total of, I believe, now $250 million in grant funding. We've put out about 100 papers. We have our first small molecule and clinical trials for oncology. And yeah, we also open access. That's maybe really, really important here. So if you have things that you want to try out, if you have a small business, a biotech, or a large biotech, we welcome you to come by and we can discuss your projects. So I'm going to stop sharing here.
Thank you, Robert. And now we're going to move on to Dr. Otto Young, and I'm going to share the slides for him. So
yeah, thank you.
Here we go.
Thank you very much for having me. I am an infectious disease physician and associate chief of infectious diseases, but my actual usual activity pre-COVID is that I'm a basic laboratory scientist, and we focus on cellular immunology related particularly to HIV infection and other immune processes. And I have to express I'm very, very jealous of my colleagues who just presented, because I have been repurposed as a clinical trials physician during this. Although my lab has been active, we've done We've done serologic assays on recovered patients. We have been collecting samples, and we will get to basic studies later. But today, what I'm going to talk about is the clinical trials I've been involved in here at UCLA, just to give you an overview of what's going on. Next slide. So this is how it's shaping up clinically in terms of the patients we're seeing. Obviously, someone is first exposed and gets infected. And it looks like there's an early phase of disease that's probably mostly related to virus-induced immune damage and innate immunity. So it lasts about 7 to 10 days. It's variable in severity from person to person. Some people can be minimally symptomatic or even asymptomatic. And then it seems like there's an inflection point after this first early phase. And some patients will then go on to recover without any intervention. And then others, severe of lung inflammation, oftentimes a systemic sepsis-like, cytokine storm-like inflammatory state. And this looks like it's probably a hyperinflammatory state overall, and maybe related to dysregulation of the immune system against the virus. And this is what leads to the significant morbidity and mortality of patients. And so looking at it this way, there are kind of 3 places to interrupt. So one is antiviral prevention of infection in the first place, or antiviral interruption of the early phase of viral replication. And then the next phase, there are immunomodulatory treatments potentially that could reduce the morbidity and mortality of the consequences of infection. So in terms of antiviral agents, which would be for the first 2 phases, really it's been very, very limited. And that's because most antiviral agents have to be virus specific because they have to interact with viral components usually. And so we've had very few things. The inflammatory phase, however, there are lots of things on the horizon because for many disease processes, modulating the immune system is part of treatment. And so there have been a larger menu of things to try for that, including IL-6 receptor blockers. Next slide. So first, I'd like to point out this has been a huge team effort. So I had never run a clinical study before in my life until March. And so I've gotten a huge amount of support from UCLA CPSI in the Office of Clinical Research, and they have basically led me by the hand to turn into a clinical trials PI. And this is the website that kind of lists, that's available for you to see all of the things that are going on in detail. Next. So this is a table that shows actually all the active clinical treatment and prevention trials currently going on at UCLA. The majority of these are inpatient trials focused on therapeutics. There are a couple here that are outpatient, either for prevention or treating mild disease. And so I am going to zoom in on a couple of these that I'm most heavily involved in. You can see that I'm PI on several of these. They just fell into my lap. And again, this is not my usual role. I'm usually a physician scientist doing clinical basic research, but have been repurposed. Next. So remdesivir, this is the one that's made big news lately. So it's a polymerase inhibitor originally developed to treat Ebola. Actually failed in that trial of Ebola, and it's administered IV. It by chance was found to have cross-reactivity against the original SARS coronavirus and MERS, now this novel coronavirus. And given this good in vitro activity, it's been tried in several clinical studies, the biggest of which is an NIAID placebo-controlled trial, of which UCLA is one of the sites, and I'm the site PI. This, as you've probably heard in the news, was recently halted after about 1,000 patients because there was a statistically significant effect. So median time to recovery was 11 days for the drug versus 15 days for placebo, which was strongly statistically significant. However, it looked like there was a minimal survival benefit, maybe, of 8% for drug versus 11.6% for placebo, borderline statistical significant. So I would conclude that this is not a magic bullet. It probably helped patients recover who were going to recover anyway, maybe did not increase survival or had a smaller effect on survival. I think questions that arise are whether it might be more effective in earlier disease or maybe even for use as prophylaxis, since the study did include a lot of very ill patients with very severe disease. The trial is going to continue, including at UCLA, and now will be expanded to be remdesivir plus a placebo plus remdesivir plus baricitinib, which is a JAK inhibitor. Next. Another exciting compound is selinexor. So this is already an FDA-approved drug, which is an exportin-1 inhibitor. It's FDA-approved for treating multiple myeloma. It's orally administered. Main side effects are nausea and then hematologic effects, so cytopenias of various types. In this trial, actually, interestingly, based on in vitro data, the dose is very low. So it's 1/5 the dose of the FDA-approved dose for treating multiple myeloma. And the theoretical underpinning for the trial, besides the in vitro data showing direct antiviral activity, is that this exportin pathway is used in the viral life cycle of many viruses, and in screening, in vitro screening for viral protein interactions with cellular proteins, there's a very strong interaction of one of the SARS-CoV-2 proteins with exportin-1. And interestingly, also the NF-kappa B pathway involved in inflammation also involves exportin-1. So theoretically, this drug could have effects on 2 different processes, including a direct antiviral effect and immune modulatory effect to reduce morbidity and mortality. The early data looked very, very promising, and the trial has just launched here at UCLA. So I'm holding my breath for the results. Next slide. And finally, I'm going to highlight RamaMap, a very exciting therapeutic agent that we're trying now at UCLA. The trial just started, but we've had a large experience using it as compassionate use open label for over 20 patients so far. It's a humanized monoclonal antibody that blocks CCR5, which is a chemokine receptor found on macrophages and activated T cells. CCR5 is used to guide these immune cells to areas of inflammation by following RONTEs and other chemokines to those areas. So you can think of it as a homing beacon for these immune cells. And the reason it was originally developed for use in humans was to treat HIV, because coincidentally, HIV uses CCR5 as a major receptor for entering and infecting cells. So this is a blocking antibody. And interestingly, it's been given in phase 1, 2, and 3 FDA trials to over 800 patients with an outstanding safety profile, which is why we felt very comfortable trying it in our patients here. And so the theoretical benefit would be reducing immune cell trafficking into end organs, including the lungs, and reducing the inflammatory damage in the late phase of disease. So there's been increasing anecdotal evidence for COVID-19. Next slide. So of the patients we've used it in here, as I mentioned, we've done over 20 patients as compassionate open label use outside of the trial. And I just chose the first 5 mild to moderate disease patients that we tried it in. And here are some lab profiles that I have pulled and graphed. So C-reactive protein is the top graph. And so the bottom axis is time. And then the The y-axis is the amount of C-reactive protein, CRP. So this is an inflammatory marker. It's been shown to be a prognostic indicator where high levels are poor outcomes patients. And so what you can see here is that of the 5 patients, 3 started with fairly high levels of CRP. And in all 3 of those, after RamaMap was administered, which is the green portion, shaded portion, there was a drop in CRP. Another marker that's used quite a bit is total lymphocyte count in the blood. So the total lymphocyte count drops in patients with more severe disease, and presumably that's because the lymphocytes are trafficking into the end organs from the blood. And so what's shown here is that after RamaMap was given, that most of the patients had marked rises in blood total lymphocyte counts. So perhaps reversing that pathogenic process. Next slide. And so our experience here is fitting with this preprint, pre-report of RamaMap by an HIV researcher colleague of mine. So this is about 10 patients treated in a hospital in New York. And what they're seeing is drops in interleukin-6, an increase in depressed CD8 cell levels, and a normalization of the CD4, CD8 T cell level. So they've done more detailed experimental tests. And so this is an exciting agent potentially for treating that later stage of disease. And that's it.
We're at about 20 minutes till the end, but I do want to talk about the collaborations at UCLA. I know, Brigitte, you had shown on your slide your collaborations with some of the other researchers, but let's touch on that for everybody. So let's start with Brigitte on your collaborations with Vaithilingaraja and Robert.
Yes, so I think that's one of the most amazing things about UCLA is that there's been just this tremendous amount of collaboration that's happening across the board. I think a number of researchers have thought about how their work can be repurposed and how it could be relevant to COVID-19. And everybody's really trying to work together. And so, so for us, in terms of thinking about lung disease and repurposing our lung models, it was a colleague who connected me to Vaiti, and Robert and I have already been working together for many years. So it was just sort of, it already came together very naturally and easily because of the unique environment that we're in at UCLA.
And Vaiti, what about you?
So we were using a simple monkey cell line to grow the virus. So, however, that is kind of artificial, right? So at that time, I was looking for opportunities. Then I learned about Brigitte, and it's kind of a collaboration, and we started working together, and along with Robert, we completed the first screen. Now we are testing those compounds in the lung culture model of Brigitte. So we are really excited for this short turnaround and collaborative.
Robert?
Yeah, it's very different working at UCLA than most institutions because you have a lot of crosstalk. I mean, IT is the dear collaborator, Brigitte and Otto. I mean, we all worked before together before COVID-19 happened. And yeah, it's very fascinating. There's also a huge amount of diversity in terms of what we do. IT is obviously a specialist and expert in DSL3. Brigitte makes really beautiful models. Otto has all the clinical aspects. And in my case, what I bring to the table is screening capabilities. And we've even started to collaborate between Otto and I on, for example, serology testing, mass serology testing, screening and of populations, trying to get people back to work, that sort of thing. And yeah, it's a good time to collaborate. It's really good.
And I know, Otto, you said that you were kind of thrown into the work you're doing now. So how is it in the new position and trying to reach out to the other researchers to help on your research?
So I kind of given you like what I did last summer for right now, but in the longer run, I do have collaboration with Vaithilingaraja. So he's got a vaccine concept, and we're a T-cell immunology lab, and we will be helping with those assessments. I have, as Robert mentioned, we've been working together on the serologies, which has been really very interesting, and I've been bringing in actually recovered patients to look at their serologies, and then the tie-in hopefully to some of this clinical trials work is that we will be getting samples on some of these patients. So the company, for example, that's doing the RamaMap study, they will provide samples to us so we can do detailed studies of regulatory T-cell subsets, for example, and B-cell subsets as well. So it all ties together in the end.
That's great, and we do appreciate all the hard work that you guys are doing, so thank you. I'm going to turn it over to my colleague Daniel, who has been fielding questions from the audience, and so I'm going to have Daniel ask a few questions.
Thanks, Marvi. This one is for, I guess, Vaithilingaraja, Brigitte, and Robert, maybe mainly Robert. When you were talking about the types of molecules you're screening, there were several different modalities that you mentioned there. How do you go about prioritizing or kind of selecting which ones may be the most effective?
Yeah, by scale, basically. So for example, when Vaithilingaraja called me up one evening and said, hey, can I get some molecules tomorrow afternoon? I need some. I said, okay, how many? And he said, well, I can get in the BSL-3, which is a little small. I can maybe do a few plates. And I said, okay, I give you something that's very high value that we know does something, that has clinical data on it, and it's already FDA approved for repurposing. If someone came to me like Brigitte, for example, and says, I would like to screen molecules, I can be a lot more generous because she has models that adapt to a much, much larger scale, right? You always start with the most valuable molecules, like for example, approved or preclinical molecules. And then you try to move into things that are a little further away from the clinics that need more work. And that's kind of the concept here. But yeah, I'm actually really excited about some of the molecules. Some of them are already potent at, what's that, Vaithilingaraja, 25 nanomolar?
Ah, yes.
Yeah, so we're very excited about them.
Well, that's great. I guess this next question is for Vaithilingaraja and Otto. You've both admitted that you didn't start your careers as expert coronavirus investigators, but that you've worked in infectious diseases before. I was hoping you could just share how the transition has been studying, I know Vaithilingaraja, Zika virus, and Otto, the HIV virus, and if that's been able to help inform your current research, or if you've learned anything and it's easily digestible by this audience.
Yeah.
Vaithilingaraja, why don't you go first?
Oh, yes. So, I've been studying Zika virus vaccine development the past 3-4 years. So during this period, we are slowly learning about the immune response and what kind of vaccine antigens and what kind of delivery system can work for effectively prevent Zika virus. So it's kind of in a point that we could use that platform to deliver any of the antigens. So that's how we got really excited in January or February. Then we quickly moved on to get those COVID-19 antigens to our platform. So I, at that time, I've been working with Otto Young last 15 years on different projects, from hepatitis C to Zika virus to, so it's a kind of a quite natural, as an immunologist, he brings a lot of expertise this project. So that's how it's kind of initiated, and we completed the preclinical safety studies. They are really good, and now we are testing the immunogenicity profile of these candidates. So we are here now.
Yeah, so it's been really interesting for me. I think, you know, this niche I fell into this niche because I'm both still a practicing infectious disease physician and a basic virologist / immunologist. And so, you know, when COVID-19 hit, there was a huge flood of companies coming in offering different things to try. So this background kind of gave me a unique perspective on which things to choose, because clinical trials are very expensive and labor intensive. So we had to prioritize and choose which things to try. And I'm hoping that, you know, longer term that there are several things that I'm working on, my lab is working on, that will dovetail. So we are, for example, working on with a company maybe to make hyperimmune globulin instead of using convalescent plasma for treatment. I have a collaborator who's going to do single cell B cell receptor sequencing, and hopefully we can isolate some highly effective neutralizing antibodies. So it's been a really interesting, probably the most potent opportunity to kind of combine my different skill sets, much more so than even HIV.
Thank you. We had another question, and I do too, for Brigitte, about the organoid model you're using and how it surpasses some of the other models and like the limitations of those. If you could just kind of help us compare and contrast the two.
Sure. I think, you know, just growing cells in 2D on plastic is obviously very different to how the cells experience their microenvironment within a tissue. And so as much as we can model what's actually happening in the body in terms of cell-cell interactions, recapitulating stem cell niches, growth factors, you know, all of those things is obviously very important and will probably much more closely mimic how patients are going to respond to therapies if we can really recreate those tissues. You know, there's some questions about how rodents are going to respond to the virus in terms of whether that's a model that's relevant or not in terms of patients. And so I think as much as we can recapitulate the human tissues, that's very, very helpful. So a lot of the current screening, which is very easy to do in 2D, may just not be that relevant to patients. And so you may find hits that look really good in the dish, but when you get it to patients, they just may not be relevant. So we're really trying to try and really find compounds that we think are much more relevant to patients by having really good models up front.
Great. And this next one is for Robert. You mentioned serological testing. You kind of highlighted, you know, the need for more specificity and accuracy. Can you just talk further about that, and I guess what needs to be done in order to safely rely on these methods?
Yeah, let me talk a little bit about the different assay types that are out there. So, for example, here at UCLA, they have assay types implemented in the clinical laboratories that are very, very reliable. So, for example, systems that are like the Abbott machine or like the Roche machine that have specificities that are approaching 100% and selectivity is also 100%. So you can really rely on these. And then there's other assay types that are just ELISA-based. That's a very old technology that basically mobilizes an antigen and just looks what from the serum of the patient binds. And you detect that with another antibody detecting the antibody of the patient in the serum. And that has its trapdoors. This means that you can get false positives, false negatives, and so on and so forth. But by and large, these are still relatively reliable if you have a clinical laboratory that is good at this. And then you have lateral flow tests, and those don't work so well. And that has something to do with the fact that they're mostly using actually the N protein, the nucleocapsid protein of the virus for this lateral flow test, where you have, for example, a serum that of blood from a finger prick that you're applying, you run it. If there's a stripes, then supposedly you actually are positive, you have antibodies against the virus, and if not, then not. So problem is number one, there's a huge homology between different types of coronaviruses. There are some that are commensal. This means that they're in the population, but they don't cause anything like COVID-19 infection associated diseases, right? They're a little bit more like the flu. And then this means, however, that if you had something like that, you might have already antibodies against the nucleocapsid protein that's very, very similar to the nucleocapsid protein of COVID-19. This means that it will look like you actually have antibodies against COVID-19. You might conclude that you are protected, but you aren't. That's a huge problem. So these particular lateral flow tests should be approached with a huge degree of caution. Also, we've seen lots of cases where actually they're negative when they should be positive. We tested something like 7, 8 of them, and we were not impressed with the performance of them. Right now, what we're doing internally is we're setting up a gold standard for benchmarking all of the tests against, so that we can make some well-informed recommendations as to where we are going with this, what we should be testing in terms of antigens, how we should be processing our samples. We're trying to make this scalable. We're trying to make this cheap. We're trying to make this really accessible to people. That's the thrust of our research right now.
That's fantastic. So we have time for one more question, and this is going to be directed towards Otto. We're getting a lot of questions on, and forgive me if I pronounce this incorrectly, asking about recent information on whether COVID is a Rantes problem. And I guess a follow-up question would be, if you can just share just the latest on the clinical trials, especially for leronlimab, and kind of what you think the I guess next few weeks or next few months are going to look like with treatment.
Sure. First, I would like to just add one more comment regarding the antibodies that Robert was just talking about. So, you know, I would add that the accuracy of the test is not the only issue. I think there's an implicit assumption right now from politicians and many people in the public that they correspond to protection. That very well may not be true. It's not true for other coronaviruses. So big caveat there. So on to the issue of the pathogenesis of the disease and whether it's a Rantes problem. So that preprint that I mentioned does seem to suggest that the level of Rantes, which is the chemokine that CCR5 recognizes for trafficking, does correlate to severity of the disease. So it may be a marker. I'm not sure that it's a Rantes problem itself, but that's probably a marker of the process that's going on of the trafficking of immune cells in and causing the inflammation. In terms of how effective leronlimab is, you know, I can just tell you, this is anecdotal evidence because we've given it to over 20 patients, but it's not been a randomized controlled study. I can tell you there have been some examples that, including one that's been publicized a lot, of some fairly dramatic turnarounds of patients. That's not been universal, so not all patients have gotten better. You know, if I were to guess, so it's really hard to know because these patients are getting multiple interventions. Some of them are also getting plasma, some of them are getting hydroxychloroquine, some are getting IL-6 receptor blockers. But if I had to guess, I would say roughly half to 2/3 look like to me like they may be responding. But again, a formally controlled clinical study is going to be needed, and that is in the works right now.
Great. Well, I'd like to thank our panelists for this engaging discussion today, and we'll turn it over to Marvi for final words.
Thank you, everyone. So again, we appreciate all of your work, Vaithilingaraja, Brigitte, Robert, and Otto, and we appreciate the time that you gave to us. This webinar will be available on video for future. If anyone needs to look at it, just take a look. I'm going to share my screen real quick with some information here. Yeah. So you can just take a look at our website for the future. And again, thank you so much, and thank you to the people who attended. And we wish you all luck and hope that we do find a cure to end COVID-19. Thank you.
Thank you, everyone.
Thank you.